share our office and lab space with colleagues from the University of Queensland, and the work I'm presenting today was done in conjunction, so in collaboration with Professor John Thomas here and his postdoctoral staff, Dr. Megan Vance and Dr. Nan Tran. So I'd like to acknowledge them up front. And we also have a wonderful team of technical assistants behind us as well. Before I launch into the scientific presentation, I thought I should introduce where I'm from. You can see I've got Bogor marked on the map, and I've also got a star for Brisbane. So Brisbane is where our office and laboratory is located. And Brisbane is the third biggest city in Australia at about 2.6 million people. So not quite as big as your city here, but similar size. We are located on the Brisbane River, and it's a coastal city, sprawling to the north and the south and a little bit to the west. Our work is located at the Ecosciences Precinct. So that's a fairly new building, about 10 years old, and it's where Vuita spent 4.5 years with our group for her PhD studies. We have, the building has capacity for about 1,000 scientists, and we have glass houses on the roof, multiple basement levels where we have boat storage for the fisheries people that go out and do fish and water catchment sampling. We have controlled environment rooms, an electron microscope in the basement, so lots of facilities there. And the idea is that we've had staff from lots of sites come together to promote the collaboration between staff and fertilisation of new ideas. So John and I have developed collaboration with our animal science colleagues who work in virology. Right, but I'm not actually a Brisbane girl by upbringing. I grew up in the north of Australia in Cairns. So Cairns is in the tropics, and I've really enjoyed coming to Indonesia. This is my first visit here. The weather reminds me of my home in Cairns, where it's warm and humid with a dry season and a wet season. Cairns is only a small city of maybe 160,000 people. It's much bigger now than it was when I was growing up there. And we talk about the tropical north of Australia being where the rainforest meets the sea. That said, it's got a lot of agriculture in the area, a lot of vegetable crops, a lot of bananas, mangoes, and a lot of sugarcane. And you can see the sugarcane crop here close to the mountains, and that's only taken maybe 2 kilometres down the road from my parents' house. So I love the tropics, and I look forward to coming back to Indonesia more times. Anyway, into the science. I know you will have all seen bananas, because I don't think I can go 10 seconds in Indonesia without seeing banana plants, but perhaps you don't realise the science that we know behind bananas. Ita, I can share the slides with you.
Oh, okay. So just take a picture of this.
Thank you. So bananas are not actually a tree. They don't produce wood. So we consider them a very large herbaceous plant. They produce a berry, which is the botanical term for the fruit. And they're grown in a lot of the tropical world. Quite important staple food crops in a lot of places. In those places, some countries produce a lot of bananas, but it's all used domestically, and other places, particularly the Americas, export a lot of bananas to the non-tropical parts of the world. While there are many, and I'm talking hundreds of banana cultivars, the export trade is mainly Cavendish bananas, which I have a friend who calls them plastic bananas. They look good on the outside, but they don't taste very nice on the inside. Unfortunately, the Australian industry is based on Cavendish. they haven't discovered the delights of all the other yummy bananas that are available. Bananas are generally infertile. They don't produce seeds. That's why we have the flesh that can be eaten. And they're infertile because they tend to be hybrids between either Musa acuminata species or sometimes crosses with Musa balbisiana. And they have either diploid or triploid genomes. There are also banana cultivars out of breeding programs that have tetraploid genomes, but they're not very common. Important to remember that because they don't have seeds, they're vegetatively propagated, and that's a great way to accumulate viruses in any crop. And the wild bananas, so the ancestors of the cultivated bananas we have, are really important as a source of resistance for diseases that we have in banana, and also the potential to meet environmental challenges, tolerance to different climates. So, banana viruses. There's a whole pile of banana viruses known. I've got some nice photos up here of a range of them. Some are important in, or are known in restricted distributions, so limited places around the world, and others are quite widespread. Important to note here that banana bunchy top virus has a very close relative called Abaca bunchy top virus. It is a distinct species, but both viruses infect both Abaca, which is Mucilus textilis, and the cultivated banana cultivars. In the world scene, we have a number of really severe threats for banana production. Indonesia has 4 of these ones. So, lucky you. The one that you're missing is Xanthomonas wilt, which is a bacterial wilt known in Africa. But you do have a bacterial wilt, the blood disease, which is primarily known in Indonesia and has spread quite rapidly in recent decades, and now appears to be spreading to other parts of the world. And you can see that bunchy top is in that list of the top pathogens of bananas. Banana bunchy top virus is the most destructive viral pathogen. In Cavendish crops, it can cause near complete crop loss within 1 to 2 years. It looks like a nutritional deficiency, so often it's unrecognised by novice farmers until it's spread quite far through their crop. Biologically, it's a low concentration virus. It's limited to the vascular tissue, so particularly the phloem, which is the sugar transporting tissues, and it's a multi-component genome. So each approximately 1 kb component of the genome is separately encapsulated. Each component encodes for a single gene. And these particles, the virus particles, are only 18 to 20 nanometers in diameter, and that makes them the smallest virus particle of any host that we know. So genetically, within banana bunchy top virus, we have 2 subgroups, so distinct variants, the Southeast Asian subgroup and the Pacific and Indian Ocean subgroup. The maximum diversity of bunchy top within the Southeast Asian group is within the Southeast Asian group, and you can see that part of Indonesia is covered by that, but you can also see on my map here on the left that the wild progenitor subspecies of Musa acuminata are also present in this area, and you can see how much of it is present as part of Indonesia, making it a really valuable source for wild germplasm. Bunchy top is present in most banana growing regions, with the exception of the Americas. And within those regions where it's present, it's still spreading. You can see the stars on the world map there. Those are detections that have been made in areas that weren't known to have bunchy top, but now are known. So, how is it spread? Firstly, it's spread through planting material. So, when suckers are taken from a clump of bananas and transported to a new site, or through tissue culture, which is common in some parts of the world, bunchy top is transmitted through that material. It's also spread in the field by very specific aphid species, so 2 species in the Pentalonia genus. In Australia in the 70s and 80s, and then using Australian data very recently, we've had 2 rounds of epidemiological modelling done that's been facilitated with computational models on really high-level statistics. We have colleagues that do that. That's not my area of expertise. But you can see with both of the models, they're giving us very similar results. And what it tells us is that bunchy top is most likely to spread to plants that are fairly close, but it is possible to spread by aphids easily out to 100 metres. However, that said, we've had examples of the aphid spread of up to 10 kilometres. So where the aphids get up into the atmosphere and they're blown a long way and then drop out into a new site. So in Australia, as I said before, we have mainly large Cavendish plantations. We have the majority of our bananas are grown in the tropics around where I grew up in Cairns. We call that the wet tropics. But we have a small amount of production in other parts of Australia, including around Brisbane, so southeast Queensland and northern New South Wales, where we also have banana bunchy top virus. So we had a single incursion from Fiji over 100 years ago in planting material when the industry was rapidly expanding. We've been quite fortunate that a combination of government regulation on movement of planting material and then industry action has kept the disease level quite low and stopped the movement of the virus or contained it. We have had a couple of examples of incursions into the north of Australia, but before we had a large industry and they were able to be eradicated quite quickly. I think if we got it into North Queensland now, we would be in big trouble. So the management that the industry undertakes is regular inspection and then prompt and appropriate destruction of infected plants and then replanting with clean planting material. The frequency of the inspections is, has been calculated based on the computational modelling, epidemiological modelling that was done in the 1970s and 80s, so that we have an evidence-based plan of action. So as I said, the disease resembles nutrient deficiency. Bunchy top virus has the effect of changing photosynthate movement within the plant, and initially you get discontinuous dark green streaking that's visible on the underside of the leaf, and I've got an example up there. But then the leaves become bunched in appearance, they stand more upright, and they have yellow or chlorotic margins that are slightly curled upwards. Normally banana leaves curl downwards. And in some cultivars, you get leaf distortion. Different cultivars have different reactions to the virus, and some cultivars, the symptoms don't appear as strong, but the end result is that the plants don't produce fruit. when one stem becomes symptomatic on a clump, the virus does transmit very rapidly through the whole clump, and eventually every stem will show symptoms. Infected plants do not recover, and some plants are harder to infect. So I had a colleague working in Yogyakarta for her PhD. She wasn't working on bunchy top virus, she was working on blood disease, but she had a patch of Cavendish bananas and a patch of Kepok bananas. The Cavendish bananas very quickly succumbed to banana bunchy top virus. It spread really rapidly through that area. The Kepok bananas did develop banana bunchy top virus, but their rate of transmission and how many plants over time got the virus was much slower. So they're susceptible, but they're harder to infect. So we're getting more deeply into the disease. If you've ever looked really closely at a banana, you might have already seen that they produce one large leaf at a time from the centre of the plant. So you can, if you're looking for the most up-to-date status of bunchy top infection in a plant, you look at the youngest leaf. Following infection, so aphids carrying the virus can feed anywhere on the plant, and following infection, the plant produces often 2 new leaves before you see symptoms. It can be a little bit longer, but more leaves under some circumstances, and we're still working on understanding why that would be. But we think maybe it has to do with the amount of virus that's inoculated into the plant. Symptoms then increase with severity with each new leaf that's produced. So, original work that was done by Charles McGee in Australia when we first got banana bunchy top virus, and it was a really big deal, found that aphids had to feed on symptomatic material to transmit the virus. The epidemiological modelling that our inspection program is based on found that the average time between a plant becoming infected and the time that the virus was spread from that plant was just under 4 leaves. So bananas grow at different rates depending on the temperature. In subtropical Australia, in summer when it's warmest, our plants produce about 1 leaf a week. So every 4 weeks they've produced 4 leaves. So that's the frequency that our inspectors go to the different properties to try and catch infections as early as possible and slow the spread of the virus. But in winter, the plants slow right down and they only produce 1 leaf about every month. So it can take longer for new infections to become apparent. So 2 things happened around about the same time. We were working with statistical modelers to develop our new model so that we could use it for forecasting and how to best use the industry resources for the inspection program. And they found that parameter of 3.7 leaves from infection to spread didn't really match up with what they had to use as parameters in their model. So they started raising some queries with us. And then in the field we were testing some new antibodies that I'll talk about later in my talk. When we got unexpected results. So to test our new antibodies, we went out to the field and we used a place with a hotspot of infection where there were a number of plants that the inspectors had identified. They had symptoms, and we sampled a lot of asymptomatic stems around those symptomatic stems and tested them for banana bunchy top virus. And the graph here shows The height of the bar is the relative level of the virus in each stem. Now we sampled the youngest leaf of each stem. Unexpectedly, we got high levels of virus in plants that had no symptoms. So that made us really wonder what was going on, and we've investigated further. Now, pause for a second. When we say symptoms, once you have a chronically infected plant, they're very obvious. But when a plant is just developing symptoms, they can be faint or mild or patchy. And we've had examples where on a whole big giant banana leaf, we've got one line, one dark green line, or a few discontinuous flecks. So we've looked, when we say that the plants were symptomless, we have looked incredibly hard now to make sure that we could see no symptoms. So the first thing we did was essentially repeat our initial finding, or initial investigation. We went out into the field, and you can see John and our colleagues collecting from the youngest leaf, and the plants are quite tall, so even tall John is on a ladder. And these were, again, from symptomless stems, and we collected from every stem in a clump, and lots of clumps around a hotspot, a collection of clumps that had symptoms. We took them back to the lab, and we were working in the COVID times, so we're all dressed up in our masks as well as our regular PPE in the lab, and used ELISA to quantify the level of bunchy top in each stem. This is what we found. We also, once we had the ELISA results, we went back and we've also spatially mapped where all the plants are, and you can see that we've got a number of plants that had no symptoms but detectable virus. And in amongst quite a number of healthy plants. We did the experiment a second time and found similar results, but not quite as many plants. So we have tried to have a look at that as well. But between our first and our second samplings, we went back and monitored those plants that had no symptoms and checked them again for symptoms as each new leaf emerged, and also quantified the virus level in the plant. So we took each colour here represents a different plant, and the height of the bar is the relative level of the banana bunchy top virus. The solid bars are leaves that had bunchy top symptoms, and the pattern bars are the ones that were symptomless. And you can see the light blue one there is what we expected based on Charles McGee's work, where the plant went from symptomless leaves with no virus to leaves with symptom and detectable virus. But you can also see there's quite a number of plants that produce a small number of symptomless leaves with detectable virus prior to producing symptoms. So, bunchy top infection numbers of infected plants in the inspections varies over time. Because the plant growth rate slows down in winter, the detection rate is slower, and then it increases as the plant growth rate increases over summer. And we did wonder if maybe the production of these leaves was temperature related. Could they vary seasonally, and it's only at some times of the year? And my hypothesis was when the plants are slowing down after the peak of summer, that maybe you get this stretched out time, and that's when we'd see the symptomless leaves, because when we did our detections it was autumn. But so we've done a lot of monthly sampling, and we've repeated that large sweep sampling a number of times in different seasons. The monthly sampling has shown that the plants appear to produce one or two asymptomatic leaves with detectable virus throughout all the seasons. This is data from plants that have produced them. It's asking how many have they produced. How often are they produced? Well, that's a bit of a confusing story. We worked at 2 different sites at the range of where bunchy top is known in Australia. The Queensland site is further north and it's a little bit further inland, so it's a little bit warmer in the day, a little bit colder at night, but the temperatures are fairly similar. We're only talking a couple of 100 kilometres, maybe 300 kilometres north to south between the 2 sites. You can see that there's a very different pattern in the occurrence of plants with these asymptomatic virus-infected leaves. One point to note is that the Queensland site had to be stopped early. The management of the bunchy top was not ideal there, and it did get out of control, and the grower elected to destroy all the plants in that paddock to try and protect the other plants that he had on the site. At the New South Wales site, the bunchy top management is much more as we would expect, but we've also had very commonly detections of this symptom or lack of symptoms with detectable virus. So our question is, it's obviously a far more complex story than just temperature alone, and we don't have the answers to that yet. The critical question though is, are these asymptomatic leaves with detectable virus infectious? Can they be a source of infection for surrounding healthy plants? John and I have done quite a number of aphid transfers where we would feed, we have a Pentalonia nigronervosa colony, so the banana aphid. We would feed those aphids on the leaf tissue that we'd tested by ELISA, had no symptoms or known symptoms, and it gave them an acquisition period of 2 days, followed by another transfer onto healthy plantlets, and then an inoculation period of at least 2 days. We sprayed them with insecticide and monitored their growth in the glasshouse. And what we found is that regardless of the presence of symptoms or not, we were able to transmit the virus where we were able to detect the virus by ELISA. So that's now being incorporated back into the epidemiological modelling so that we can make some updated recommendations to our industry management. And that'll be important if we have an incursion of the virus into North Queensland as well. So because I suspect our biosecurity agency will want to mount an eradication campaign or determine if it's feasible. And it's also got relevance to lots of other parts of the world where management of bunchy top is important for food production. I'd like to jump onto some different research. It's related, but something that in John's Bill and Melinda Gates Foundation project has been worked on is the development of an in-field assay for the virus. So the symptoms are really obvious when they're severe, but not everyone is familiar with the symptoms. Or it could be really, really important that the material is clean. Say if planting material was being distributed from the site, which I believe is what happens in Africa. The existing assays are based on serological tests, so ELISA, and we use immunocapture as our template preparation for PCR. Because they're low cost and high throughput, we could certainly do a DNA extraction and a PCR, but that adds a lot of cost and a lot of time to the testing. The problem was that the antibodies that we used for the tests had reached short supply. We weren't able to get any more. So our colleague Megan Vance worked with us, and we created new antibodies, both polyclonal antibodies that came from rabbits that were injected with purified virus from infected plants, and we injected 2 mice with a similar preparation, and then monoclonal antibodies were generated from those. So the process of doing that is and we outsource this, we don't do the animal work. The mice are injected and they receive a series of doses, and then after the final dose, and we checked a test bleed that there'd been an immune response, the spleen of the mouse is dissected out of the mouse. Those cells are then fused with a myeloma cell line, which gives it perpetual life, and you're creating a hybridoma cell line, so perpetual life and the ability to produce antibodies. Each cell line produces only 1 antibody, but we had lots and lots of cell lines to screen. In the end of the screening, we found 150 lines that were worth pursuing, and we've ended up with 26 monoclonal antibodies. The top 2 are what we've taken forward for this. But each antibody is specific to a different epitope, which is a different part of the virus. Right, so what we did with them, as well as checking that they worked in our existing formats, and they're great, Megan has developed a lateral flow assay, otherwise known as a rapid antigen test. We're all really familiar with them after COVID. So the way the test works is that you put your potentially virus-containing sample onto the sample pad. The virus then binds with antibodies to the virus that are bound to gold particles, and everything flows up through a series of points along the strip. Those potentially virus particles bound to the antibodies on the gold particle then interact with 2 strips of antibody lines. One is a line of antibodies that will react to the mouse it's an anti-mouse antibody, and remember our bunchy top antibodies were generated in a mouse. But you'll only see it if you've got the gold particles accumulating there. And then the other, that's your control line, and the other is an anti-bunchy top antibody, that's your test line, and it will give you the positive if you've got the virus particles bound to the gold. I think I've made that a little confusing, but would you like me to do that again?
Yes.
Okay, I'll start my head again. That's a good idea, John. So, the pointer doesn't work on there, so I'll be clearer.
Yeah, you can.
Yep. Our plant sample comes in, and the idea is that the virus particles in the plant sample interact with antibodies to banana bunchy top virus that are stuck on the gold particles. So we call that the conjugate pad because the antibodies are conjugated to the gold particles. Everything then moves up the strip, and where you've got virus particles bound to the gold particle, the line of anti-bunchy top antibodies will mean that the gold particles stick there. The excess, ones that don't stick, because not everyone will stick, flow all the way through, and they get captured on the anti-mouse antibody line. So, is that clearer? You end up, the lines you can see are because the gold particles are sticking. They stick depending on whether they have virus bound to the gold particle or not. Better? Okay. Have a think about it. I'm happy to answer some questions later.
Sure.
The thing to take home is to develop one of these tests is there's an awful lot of optimization that has to happen. Every different component you can think of, concentrations of antibodies, material that's used for the sample pad and the conjugate pad and the buffers and all sorts of things, including even the types of gold particles, all had to be optimised. But at the end of, I think it took Megan most of a year, we ended up with 2 lateral flow assays based on different sized gold nanoparticles. And it's properties of the gold nanoparticles that give us the different colours. So you can see that for both of them, we only get a colour in the test line when we apply infected, so bunchy top infected sap, and we don't get anything in the healthy samples. These tests are sensitive. They're at least as sensitive as ELISA, although, hang on, I'll go back. So we have the green ones, which are based on the larger gold particles, and the red ones are based on smaller gold particles. And here we've just presented the results for the larger gold particles, which seems to be more sensitive, but they're both within a reasonable range. So they're sensitive, they're specific. We've tested them both against a range of banana viruses and also a range of banana bunchy top virus isolates from different places, representing both of the different subgroups. And we get detection where we expect to get detection and no cross-reaction with other banana viruses. We've also tested it against a related nanovirus, so a close taxonomic relative to banana bunchy top virus, and got no cross-reaction. We've even tested it in the field, in the back of the ute, in the middle of the banana paddock, and got a result within just a few minutes. So easy to use. And this is some of our team of helpers on one day when we were out in the field. Right, I've talked a lot. I'm sure you've got some questions.
Thank you so much, Katie. It's very interesting lectures. So hopefully students also have many questions. I'm sure in their heads already had many questions. Okay, in the first term, I will open for 3 questions first. And then, are you happy with that? Yes. Okay, please raise your hands. If you are said to speak in English, it's okay to speak in Indonesia, and I will translate. Ayo, ada yang bertanya? Andri?
What would you like to ask?
Okay, thank you. Good morning. My name is Andri Saputra, and I will ask you, what is the host range of BBTV in Australia? Because I think in Indonesia the host is just in banana, and from the 2 factors between Pentalonia nigronervosa and Callidii, which one is more effective to spread the BBTV? Thank you.
Thanks, Andre. That's some really good questions and touches on other work that I haven't presented here today. Okay. Up until very recently, worldwide, people were under the impression that the only host of banana bunchy top virus, was in the Musa genus, and I think the Enseti, which is a related genus. However, in recent years, there's been some definite detections of field infections in limited alternative hosts. And John and I are very keen to do some more work on that. In Australia, we haven't had any of those alternative hosts identified as infected with banana bunchy top virus. Back in the late 90s, John and our colleague Andrew Gearing undertook a lot of experimental transmissions with Pentalonia nigra nervosa, trying to get bunchy top from banana into a range of different host species, related hosts, and were not able to do it. We think that was Pentalonia nigra nervosa because it came from banana, but since then there has been a taxonomic split. So Caladiae and nigra nervosa, the two Pentalonia species, were originally considered different forms of the species, but they've now, I think it was 2012, were recognised as separate species with host preferences. We've tried to repeat the inoculations with a Pentalonia caladiae colony and weren't able to do that. Now, one of the explanations could be that our isolate of banana bunchy top virus doesn't infect non-banana hosts, but perhaps other isolates overseas may do. We know we only have really limited genetic variability in our banana bunchy top virus in Australia. So we think a single incursion. What was the rest of the question? I know I'm missing bits. Yes. Yes. So overseas, in different parts of Asia, and we know in, for instance, French Polynesia and Hawaii, there have been identifications of field-infected hosts. Something I'm hoping to explore is the genetic differences between those isolates. That how did they gain? What genetic changes represent the gain of host range? There's a lot of questions around that. And then does it become a big problem or not? In French Polynesia, they certainly have a large problem with banana bunchy top virus infecting an Alpinia species, which they use for cut flowers. And we're working with them to characterise that situation so that they've got some advice. Prior to that, they didn't have banana bunchy top virus there. So there's the question of how widely that could be spread in the Pacific. But it's certainly an open area of research. The host range of the two species of aphids may also be a contributing factor, because while you can find both species on banana and banana relatives, it's extremely, well, it's rare. I won't say extremely rare, but it might be 2 or 3% of the time. What you pick up is the non-preferred host. The aphid that doesn't prefer that host. That could be a factor in transmission between the alternative hosts. Have I covered everything now? Maybe John will.
Just make one more comment about that, in that, well, there are definite alternative host records from Indonesia, and some from the Philippines, some from Taiwan, but I've collated all that information for the people who have found the virus or not found the virus in alternative hosts, and for virtually every record where someone has found an alternative host, someone else has done experiments and not been able to infect it. So there's plus and minus infection for all the different hosts, alternative hosts. So we don't know whether that's the isolate of the virus or the subgroup of the virus, or whether it's Nigra nervosa or Caladiae, or whether it's even different strains of the plant. So that all those factors together might mean sometimes it's important and sometimes it's not. And we also don't know how epidemiologically important it is, because sometimes the infections are natural, as Kathy mentioned in French Polynesia, but many times they're artificial inoculations in the glasshouse that we don't know whether it happens in the field or not and whether it's important. So it's a big area of unknown. So there's both species. Oh, you mentioned transmission efficiency too, didn't you? Did you want to mention that?
Thanks, John. Yeah, both species are definite vectors. And our work using both species to transmit banana from banana to banana, they were similar efficiencies. Yeah, so it could be, again, different lines of the insect, different subgroups or specific genetic details somewhere in the system. Good questions.
Okay, Andre. Okay, next question, please. Raise your hand. Tiara, you want? Ada pertanyaan, Tir? Okay, Purnama has a question.
While waiting for students to have a question. Yeah, in your presentation, I saw that the biggest production banana is sold out like in India or something like that. Can I see the slide again?
Oh, that's right at the start. Yes.
Yeah, that's one. Yeah, just wondering, is it that area is in India, the biggest production?
Yes.
Okay, it's just related with biodiversity of banana too, or only the production? I'm just wondering about that.
I think it's just production. I didn't make this figure. I can't remember where it came from, but yeah, it's the amount of bananas that are produced.
Okay.
And it's
Yeah.
obviously got to be somebody's estimate. Nobody goes around and counts every single bunch of bananas.
Yeah, I'm just wondering what the big countries in the world that have the most biodiversity of banana varieties. Indonesia? Oh, okay. No, I know. Okay. And another question actually. You mentioned about the symptomless and symptomatic plant, right?
Yes, and leaves.
Yes. I'm just wondering, is that also influence the productivity? The symptomless, for example, is still higher productivity compared to symptomatic, for example.
They only produce, the plants with these symptomless infected leaves only produce 1 or 2 leaves like that before they then produce typical symptoms. So the virus then stops the production of fruit by the plant.
Oh, still stop the production
Yes.
of the fruit. Okay.
Yeah. The big implication is that the spread of the virus is likely to be happening much earlier than we thought.
Okay.
Which then means the epidemic spreads more quickly and accelerates.
Yeah.
Or we have an explanation for why it's accelerating faster than we expected. Another reason for this work was that on most properties in Queensland and New South Wales in Australia, the inspectors can keep the bunchy top under control. Sometimes they eradicate it and get the numbers down to 0, and then they get reinfected. But everything's generally at a low level of infection. However, we've had these 2 properties that we worked on where the infection got out of control, and particularly the New South Wales site. They've been working there for 6 or 7 years and doing what should be effective to control the epidemic and reduce the numbers, and it's not working. So the big question from industry is why, and therefore what else could be done to better control the problem. Because when you have a large source of the virus, then it starts impacting on other properties as well.
Yeah, so Kathy mentioned during the talk, the epidemic, the control didn't fit, the control methods should have worked, But the epidemiological modelling work that was done showed that spread should be occurring, looked like it was occurring much more quickly than everybody previously thought. It looked like it was spreading after the plant had produced 2 or 3 leaves after infection, not 4. Yeah. And then Kathy's work has shown that we can transmit the virus earlier than we thought. So that goes back and supports the new models. And one reason might be that in a plantation like the New South Wales plantation, it wasn't very well managed. There are a lot of dead leaves on the plant, lots of suckers, and lots of aphids. So we think there's a lot more transmission happening, a lot more chance of transmission happening. And as Kathy's shown, we can transmit from leaves before symptoms appear. And if there are lots of aphids, the chance of that happening gets more and more likely. So the epidemic is spreading quicker. And one other point is, Kathy's describing leaves that are produced on a plant before symptoms appear, but there's detectable virus, but all of those plants eventually develop typical symptoms. We don't have plants that never have symptoms. They've all got symptoms. It's just a matter of when we can detect the virus and when we can spread the virus.
One last question. Within the comment is very susceptible, right? I just wonder if there any special thing that makes covering this very susceptible, like certain gene receptor or something like that. Because I saw in particular, the one area is all infected, but other varieties is fine.
So the question was, is there something that makes Cavendish bananas particularly susceptible to banana bunchy top virus? So Cavendish bananas are a AAA genome, and work in John's project has identified likely sources of resistance from the B genome. So Cavendish lacks those sources of resistance. The added complication could be that there are factors that make the banana more palatable to aphids, or the right sort of feeding that would lead to virus transmission. John, would you like to elaborate? We're starting to move out of my particular depth.
Yeah, as one of Kathy's slides near there showed all the different subspecies of Musa acuminata. So yeah, the A genome is not the one thing. So all the different cultivars of banana have mixtures of all the different subspecies, and sometimes they've got Balbisiana in them as well. So all the cultivars are a little bit different from each other. And as Kathy mentioned, the Balbisianas, many of those are resistant to bunchy top, but also some A genomes have resistance, a different sort of form of resistance that we don't know much about. And Cavendish doesn't have that. Gros Michel does, and that's very difficult to infect. And the other major point with Cavendish is that people often grow large areas of it. So it's very susceptible, and there's a lot all in the one place, so the epidemic spreads. But if you have mixtures of bananas, and a Cavendish here, and then all local varieties that are not as susceptible, and then another Cavendish, the aphids don't get there and carry the virus. So, yeah, and that's a big area where a lot of work is still needed with the resistance, because we're sure there's resistance in the local Indonesian subspecies of acuminata. And ASTI's starting to work on that, but there's a lot to do. Yeah.
Thank you, John. Thanks, John.
give me an insight as well. Okay, thank you, Pak Pur. Okay, next question, please. You can raise your hands.
Peter, is there anything online?
Oh yeah, Farhan, could you please check, is there any question through YouTube? Oh, Tiara has a question.
Okay, good morning.
Good morning.
My name is Tiara. I just want to know about BBTV is present in most banana growing region worldwide. But why America not yet report about present BBTV? And how management banana bunchy top in Australia? Thank you.
Okay. So the first question, why do the Americas not have banana bunchy top virus? Well, we understand that bananas and banana bunchy top virus originated in Southeast Asia, and then over the last 4,000 years, people have transported their favourite bananas to different parts of the banana growing world, and in many cases they've taken bunchy top along with that planting material. John might correct me, but I suspect transfer of bananas to the Americas happened perhaps when there was knowledge of the presence of the virus, or perhaps they got lucky and only took uninfected material there. Certainly in more recent times, they would have had knowledge of, not bringing material in that had disease symptoms. Did you want to add anything?
Yeah. We've done some work recently looking at the genomic sequences of hundreds and hundreds of bunchy top isolates from around the world and doing evolutionary studies on it. And it looks like there's probably only been about 13 movements, incursions of bunchy top around the world from one area to another big area. So it doesn't happen very often. So in 13 times, as Kathy said, in the Americas, they might just be lucky. In Australia, it happened once. We know just once, 100 years ago. And even within Asia, it's only happened a few times. It's only got into Africa, we think, twice. Yet it's spreading everywhere in sub-Saharan Africa. But it's spreading internally. It only got into the whole continent once or twice. So probably the same with the Americas. They're just very lucky, and it's one of their major quarantine concerns because they grow so much Cavendish. They grow the most susceptible cultivar. Yeah.
Oh, the second part of your question was about management of bunchy top. Okay, so both the Queensland state government and the New South Wales state government, so each of they would be like your provinces? Yes. They have put in place legislation that restricts people from moving bananas outside of the banana bunchy top infected zone. So there's movement controls. There's nobody standing at the road checking every car. So some of it is down to luck and community education. But the other thing the industry is doing is keeping the level of the virus in those zones as low as they can to minimise the chance that somebody has a plant that's become infected that they then move out of the zone. The industry has a small team of inspectors that go round to all of the commercial banana farms in the bunchy top zone every year, at least every year. So farms are categorised by no bunchy top, known at present or ever. Farms with a little bit of bunchy top, farms with a moderate amount of bunchy top. And depending on how much bunchy top they have, that determines how frequently the inspectors come, because it's not possible to get round to every property every 4 weeks. We don't have enough staff or the funds to do that. So what they do is they target where they go, and the properties that have no bunchy top known get visited once or twice a year, whereas the ones that have bunchy top on them get visited frequently. And there's a list. When they attend, they walk through the property and identify the infected plants. The infected plants are injected with a herbicide to kill the plant, but they're also injected with an aphicide to stop aphids that are on that plant moving off the dying plant to surrounding healthy plants and carrying the virus with them. So that's a really important part of the destruction. It's one of the things that wasn't being done on that property. I said the bunchy top got out of control. They were killing the plants but not treating the aphids on the infected plant. And so the epidemic exploded. There's been the early computational modelling looked at whether there was any benefit or how much benefit there was to removing healthy plants around an infected plant. And what that found was you were actually destroying too many healthy plants that weren't infected and leaving the grower with nothing to produce from. So, and that better or good enough control of the disease was found by the regular inspections rather than destroying large areas. The inspectors then do make a point when they've done a destruction, of when they come back 4 weeks later, of checking how well the destruction has happened. You likely wouldn't see it here in Indonesia, where it's nice and warm. The plants would respond really rapidly to the destruction chemicals. But in our subtropical region, in winter, it can take 3 months for the bananas to die because of the herbicide. So we've looked into how quickly the aphicide stops aphids transmitting the virus. In summer, the plants die in 3 to 4 weeks, and it's only a small number of times, 2 days after injection, that the plants are still a source of banana bunchy top virus, and then the aphicide stops the transmission. In winter, we've had transmission at 1 week, but not any later than that. So the answer is that the insecticide very quickly distributes within the plant and takes it out as a source of infection. We don't get 100% kill with our insecticide, so what I suspect, having read up on EPG studies of related insecticides, is that we're stopping the feeding that allows the aphids to acquire the virus. Did you want to add anything further, John? Oh, yes. So the other thing the industry's implemented since about 2009 is digital data capture. So they walk around, they carry their mobile phone, and it has a special app on it. In the old days, they used a special, device in the field, but phones with apps is very easy. And they collect the GPS location of every infected plant. So they log every inspection where they turn up to look at a property, but they actually log the specific location of every plant and the number of symptomatic leaves that are on it. They then confirm their details of what chemicals were applied and the date that was applied. That's the modern data that's actually been uploaded into the current statistical modelling that we're doing with our colleagues at the University of Cambridge. So we now have nearly 15 years of that resource, and it's been incredibly valuable to use and provide evidence-based recommendations to the industry.
And one very valuable part of that data is, as Kathy mentioned, the bananas produce one leaf at a time, and we know how quickly they grow at different times of the year. So if the inspectors, if we know what time of the year the inspector looked at the plant, the date, and then we know how many leaves had symptoms, we can go backwards and we can say that plant was infected at that date. So we know not only when it was found, but we know when the aphid infected the plant. And so we can put that into the epidemiological models and see how quickly the disease is spreading. We know exactly the timing of it. And that makes the models much, much more accurate. And that's data that the inspectors are collecting all the time.
Thank you, Kathy. What's the, Kathy, one additional question. What is the apps, Kathy? It is accessible for public or for industry?
The industry pays to use a particular app. The app is called Connect, K-O-N-N-E-C-T, and it was developed by colleagues who are based in Australia and do pest and disease monitoring. In fact, one of the fellows worked on the banana bunchy top program for a while.
Wow. Okay, thank you. Okay. Another question in the second term, please. I will open two or three more questions. Ada yang mau nanya? Gak apa-apa. Mau. Oh, one question.
Good morning. My
Morning.
name is Disa. I want to know about the reason how infected plants don't recover. Is that possible if someday we can find a way how to treat this BBTV except killing the aphids and throwing some infected plants? And the second question is the last two characteristic symptoms. Why some cultivar harder to infect but also there is no resistant cultivars? I didn't get the point. Thank you.
Okay, thanks for asking your questions. Oh. Yes. Typically, in plant virology, plants they can mount a response. Not all plants, not all times. It's very situation dependent, but you can have an initial response and of the plant defense system, which neutralizes the virus, but often that results in local cell death. So you get cellular infection, but then the plant effectively kills the cells and stops the virus becoming systemic. Spreading throughout the whole plant. I'm not aware of examples where plants have routinely successfully cleared a viral infection once they have it. My understanding is plants tend to become infected and then stay infected. I'll come back to that. The effect of banana bunchy top virus seems to be to disrupt photosynthate, so sugar movement, through the phloem. And I suspect that just means it slows the plant's growth rate right down and it doesn't have the energy to produce fruit that would be marketable. Nice big, you need lots of big leaves to support the production of the fruit, and bunchy top plants don't have big leaves or ones that are photosynthesizing very well. What can happen, when I said plants don't clear a viral infection, what can happen, and I'm thinking of banana streak virus as an example, is where the titer of the virus can be high at some times, and then the plants mount a defence response and suppress the infection, and the titer goes right down. The defence response turns off because there's always a cost to whatever molecular investment is being made, but that then allows the virus titer to go back up. So you have this cycling, and in banana streak virus you have leaves with lots of symptoms, and then the next leaf might only have really mild symptoms, and so you alternate or fluctuate between levels of symptoms in different leaves or parts of the leaf. John, do you know of examples of plants routinely clearing a viral infection? Okay, so John's shared that Isla viruses can often be lost from plants, but they're fairly different from, it's rare and they're different from banana bunchy top virus. What I was going to suggest is I know people are very keen on things like RNAi work, which is a way to engage the plant defence system with a specific target. Our industry is interested in looking at that, but I'm not aware of commercial examples yet of that technology, so it remains to be seen. I might hand over to John to explain about why some cultivars of bananas are more difficult than others to infect when there's no resistance in commercial cultivars.
Right. We're talking about degrees of, or gradations of difficulty all the time. You can make the statement that no commercial bananas are resistant or immune to banana bunchy top virus. That's probably true. But if you have 100 plants of a cultivar and you infect one of them, you can say that cultivar is not immune because you got one. It's very, very useful, but it's not immune. So that's where that statement comes from. To infect the bananas, there are many different factors. It's how well the aphid carrying the virus will feed on the banana, if it likes to feed on the banana, how well it can feed on the banana, whether that particular cultivar is a good host or a bad host. It can depend on what clone of the aphid you're using for the transmission, because different clones have different efficiencies. What else? Oh, I can't think. But there are lots of different factors. The virus might infect the plant, but it moves very slowly in the plant, and it takes a long time to move to the meristem, where the growing point is, and then for new infected leaves to be produced. The plant, a cultivar like Gros Michel, it's a AAA cultivar, a cuminata genome. When a plant's infected, it has very obvious symptoms, but when you try to infect it, it's difficult. You don't get many infected plants. When you do get an infected plant, it takes many new leaves before one has symptoms, but eventually it shows strong symptoms. So that's probably poor virus movement in the plant and difficulty in the aphid infecting the plant in the first place. You have all those sorts of combinations with different bananas. If you have a cultivar that has part B genome in them, and many edible bananas have got B genome, they might have a resistance gene there as well, and that might limit how quickly the virus moves or whether it can replicate very well in the cells. So there's many, many factors, and we just don't know the molecular basis of any of it yet. With the Balbisiana, we've shown that those plants seem to be, many accessions seem to be immune to the virus. No matter how many times you inoculate them, for how long, you get no symptoms and you can't detect the virus. So that looks very good. And at the moment we've just done some crosses between susceptible and resistant Balbisianas, and hopefully when we go back to Australia we might have the first results of that to see what the inheritance of resistance is. We have no idea how many genes are involved even with that. So that's natural resistance. And with RNAi, there's a lot of work done in glass houses that shows that it has some effect, but all the field work so far shows that it's still not as effective as it seems to be in glasshouse trials. So Cavendish that have been have used genetically engineered with what is it? Constructs from DNA M and DNA S, I think it is. They look like they're very good in the glasshouse, but they still get 40 to 50% infection in field trials. So there's some hope there, but we don't know why it doesn't work as well as it should do. And nothing's grown commercially with genetically engineered bananas at the moment.
Okay, thank you, John and Kathy. Okay, last question before I close this guest lecture. Monica.
Thank you, Kathy and John. I'm Monica, and it's such a great presentation and such a great insight for us about BBTV. And I am looking for your really computational epidemiological, because I'm waiting for the real that we can use the computational epidemiological that you are creating right now, right? And it and also for the last presentations of your presentations about LFA 150, and has it commercially or not, because I think it will be easy to use in Indonesia, because talking about BBTV that you have told and you explained, we need to really taking care of the plants and really know, really monitoring the plantation, so we can know how and when the virus will infect the plant, and what will we do to manage the plant so our plant will be free from BBTV. Okay, thank you.
Thank you. Good questions. Yes, I apologise. I should have said at the end of my talk that the lateral flow assay is moving towards commercialisation. John and Megan are discussing that at this present time with the university's commercialisation pathway, with the aim that it will be available for as low a cost as is feasible. Not sure what the time frame for that is, but we're hoping in the near future. And yeah, there's a lot of people interested in using those tests. Whether they will be cheap enough that you could test every plant in a plantation multiple times, I'm not sure. It could. I see them as a use for verifying an infection where you're not sure if the symptoms are right. You could get an answer on the spot, or if you need to be really confident that you're moving healthy material, but it'll certainly support people who aren't really confident in what the symptoms are, making a diagnosis with their plants. Do you want to add any more to that, John? Okay. Remind me the first part of your question.
About the epidemic.
Oh, the
yes.
modelling, yes.
The model.
Okay, so at this stage, I'm not sure what the availability of that modelling will be to the general public. What's being developed is using modern statistical methods, and it can forecast what is likely to happen. So it's both a temporal over time and spatial, geographic model. But I think at this stage they're still getting the modelling parameters sorted out. It doesn't produce pretty graphic outputs, just graphs saying how quickly the epidemic increases or not. It was initially based on the Australian inspection data, but it's also had African data incorporated into it, so that's going to expand the range of cultivars and the environment that it's applicable to. I don't know how people who haven't contributed to the development of the model may get access. That's certainly something that we would have to be up to negotiation with the scientists in Cambridge, as well as the people that supplied the data. Because it was initially started with Australian banana industry development money, but has then been supported by the Bill and Melinda Gates Foundation money. It's certainly going to, I suspect there'll be pressure for it to be available to a range of parts of the world where it could be useful. I think you should elaborate on that, John.
The value of that modelling, these models, is that you can look at all the epidemiological data you have, put it into the model and see two things: how quickly epidemics will occur if there's no control. So if you introduce a virus into a new area and you know what the density of bananas are and what types of cultivars, you know how quickly and where it might spread. It's one thing. And the other thing is, if you've got a control system, like we have in Australia, or if you propose a control system, you can look at how the virus will spread without control, and then if you control, like you eradicate plants or you use new healthy planting material regular basis, how that will change the epidemic. So then you've got guidelines of how you should control the virus under certain conditions, and that's applicable to anybody growing bananas under those conditions. And they've done something like that in Africa, in Malawi, some of the data in our project. They've shown if you start with if you've in an area where there's bunchy top everywhere, you clean an area up, you remove the bananas and then you start to replant them, you can use planting material from that replanting. As long as you look at the symptoms, make sure you're using healthy looking planting material, you can just keep on replanting with material from within the crop. And as long as you're good enough at inspecting and you check often enough, you keep the virus level down to a level that's satisfactory. You might have 5% infection, but you still get 95% of the bananas producing. So that sort of thing you can work out from the modelling. So anybody growing bananas under those circumstances could use that sort of strategy, and we know it works. Yep. Yeah, we're looking at tropical situations. It was all developed in the subtropics to start with, and a lot of those factors are uniform across all banana growing areas, but some things like the rate of banana growth in the tropics on average over the year is quicker, and the spread in different cultivars is different. So we're feeding all that sort of information in, and we're working with people in Benin and Nigeria, as well as Southern Africa. And we're interested in the tropical areas too, because our major banana area is North Queensland, so that's much more like Indonesia. So we want to know how quickly it spreads. So anything we find out for North Queensland will be applicable to Indonesia as well. Yep.
No, I don't have comment, Pak Pur. Oh.
I will probably use Indonesian. Jadi untuk model itu kita perlu, setahu saya nih perlu epidemi data juga dari Indonesia. Jadi nggak bisa model yang dibuat di Australia digunakan di Indonesia langsung. Jadi kita harus koleksi data juga. Kemudian biasanya itu series data beberapa tahun. Baru nanti itu bisa dibikin model yang mendekati ini, kemudian dites juga keakuratannya. Jadi memang nggak bisa langsung digunakan. I just explaining that the model from Queensland, Australia, we just have different climates and probably growing system with different with Indonesia, so that we need series data of epidemic data as well as climate for several years. Then we can make accurate model. Otherwise, the model is not accurate to Indonesia. That's what I am explaining to students. Okay, terima kasih.
Thank you, Pak Pur, for your further explanation. Probably before I close this guest lecture, because I have checked in YouTube and no question in YouTube. Okay, Bu Asti, probably you have a short statement or short closing statement.
Okay, thanks, Ita.
Oh, thanks, Kitty. You can have a seat.
And it's really lucky for us, fortunate that we have special guest lectures today by Kathy and John. Well, Ita, before I close this guest lecture, I have one quick question for Kathy, and I believe this is very important for students whom they just, I think most of them are going to start their research for thesis, for a master, PhD, and also undergraduate. And most of their work will start in the field. So they will go to the field, observe diseases, and take sample from the field, and then they will bring the samples to the labs. So do you have any, recommendation how to, treat or to handle the samples from the field to the lab, so they will use the samples very well for their research? Because sometimes you have to take samples from long distance, and it's not easy, to preserve or to keep samples fresh, something like that. Do you have any trick, or
Okay. Field sampling. Can you hear me? Yep. Thank you. Right. Well, when we go in the field, and I would recommend the same for you, is you need to be well prepared for a start, and you need to have good records of the material that you collect. So we go into the field with data sheets to collect all the information, the host and the symptoms, the location, and precisely what you've done. We take lots of photos in the field as well, so that we have good documentation. It's no point bringing beautiful samples back from the field if you don't have the information about why you collected them and why they were relevant in the field. We take lots of our sample bags. Ideally, we prepare them before we go, so you're not labelling in the field. But sometimes you do have to label in the field. You find unexpected things. When we collect our samples, we put them into a cool space. So with virus samples, it's really important we don't freeze the tissue, because frozen cells degrade the nucleic acid and proteins that we're going to need for detecting the virus. But if you keep them really hot, you're also going to cause degradation to your samples. When we go, we have an esky, an insulated box with freezer bricks in the bottom. If you put your sample straight on there, you can get cold burn. So we put a tea towel, a thin towel, on top of the bricks so that we have a cool space to keep the samples happy, in good condition. Our field sampling, we've been able to go directly back to our laboratory and put them into a 4-degree storage space when we get back. But when we do longer trips or trips to further away, we can preserve them in the field. And we do that by cutting after we've done all the photos and everything, we cut them into small pieces, wrap them in a thin piece of maybe tissue. If you take a tissue and separate it into the very finest layer, and we put that in a tube with silica gel so that you end up with dehydrated leaf tissue. That will store for a long time. It stores longest the colder it is, so longer at -20 in a freezer than at 4 degrees, but 4 degrees is better than at room temperature. In our lab, we actually have a database of all the samples we collect, and all the samples that are verified virus isolates, so that we can go back to that material and know all the details about its collection and the diagnostics that was done on it. Is that the sort of information you were hoping for? The other thing I would say to students starting a research career is when you're doing a project, keep in mind the research question you're trying to answer, and then make sure you've got good controls so you can ask, is this experimental treatment any better than current practice or doing nothing? You want to keep the big picture in mind as well as all of those precise details, and think about things before you go to the field so that you are as prepared as you can be, and ask people who are more experienced than you for advice. Once you've thought about it, you can say to somebody, what am I not thinking about? What else do I need to address? Because the better your plan is before you go, the better the outcome of your whole research work.
And one very practical thing is make sure you label your samples properly and uniquely. Don't label them 1, 2, 3, 4 today, and then tomorrow you label them 1, 2, 3, 4, and your colleague labels them 1, 2, 3, 4, and you get back and you don't know what is what. It sounds silly, but that happens all the time. So every sample has a unique label, and you use it in all your lab books. And if you give it to your colleague, they take the number and the label with it as well, so everybody knows what you're working with. Yep.
All right, thanks a lot for the advice. I think we can learn, and we can start to apply this in our daily basis. Okay, John and Kathy, we are very happy today because we were able to hear all the story about banana bunchy top virus in Australia. I know this is only a small part of your banana stories in your work, but hopefully when you come to Indonesia, to Bogor again, maybe in the next year or maybe just next 2 months, or we will for sure ask you to again share some, insight to give the students motivation. Yeah, especially in the field of viruses, because I think it's getting less and less students interested in the virus research. So hopefully by inviting virology around the world to this university, I hope the research on the virology will, develop again. So again, thank you very much and very appreciate for your sharing.
A pleasure, Buasti. Thanks for having us. And lovely to see so many interested students.
Thank you, Kathy, John, and Bu Asti, and all of students. So before I close this guest lecture, please don't forget to fill the the attendance list. So this is the link, and you can fill the form, and you will get the certificate through your email. So through the YouTube or offline, please don't forget to fill the form through this link. And okay, before the next one, before I close this guest lecture, Bu Asti will give the certificate and a little bit give to Kathy. Thank you so much for your for this sharing, Kathy. tiba-tiba diganti yang tadi. So they can bring to ESP and share in the tip cream.
Yes, yeah, okay, wonderful.
But for this bread, I think John can eat quickly.
Oh, okay.